In this video, I will demonstrate the basic concepts of electron transport chain, that why electron transport chain is essential and how oxidative phosphorylation it will take place. But before discussing this topic, we must know that what is the purpose of electron transport chain. Uh, all the basic compositional elements of food, that is to say carbohydrates, proteins and lipids, after they are digested into the body, they are converted into their basic compositional elements like carbohydrates. After complete degradation, they are converted into glucose. Proteins are converted into amino acids, and lipids are converted into fatty acids. All these basic compositional food elements, they are finally converted into acetyl CoA. So we can say that acetyl CoA, it is the final degradative product as a result of metabolism of all types of main food elements or compositional elements of the food. So this acetyl CoA, it then enters into the TCA cycle, and as a process of this TCA cycle, NADH and FADH2, which are reducing equivalents, are produced. These FADH2 and NADH, they enter into the electron transport chain to produce ATP. Now, there are two sources of NADH in the body. Mitochondrial NADH, we know that mitochondrial NADH, it is produced as a result of TCA cycle, it is produced as a result of fatty acid oxidation, especially the beta oxidation of fatty acid, and extra mitochondrial NADH. The main source of extra mitochondrial NADH is glycolysis. So before discussing the basic concept of electron transport chain, we must know that how NADH, which is produced extra mitochondrially, especially into the cytoplasm of the cell, how it will enter into the mitochondria. So there are specialized shuttle systems which are involved in the transport of the NADH from cytoplasm into the mitochondria. There are two types of shuttle systems. This is the step of the glycolysis, which is involved in the production of extra mitochondrial NADH. So there are two types of shuttle system: malate aspartate shuttle system and glycerol 3 phosphate shuttle system. The malate aspartate shuttle system, we know that malate, it is produced as a result of the reduction of the oxaloacetate, uh, which is produced by the pyruvate into the cytoplasm, and this malate oxaloacetate is reduced into the malate. Then with the help of specialized shuttle system, which is called malate aspartate shuttle system, malate will enter into the mitochondria. Now basically this malate is formed from the oxaloacetate with the help of enzyme malate dehydrogenase. As a result of this activity, what happens that NAD is reduced to NADH. So we can say, NADH is oxidized to NAD. So hydrogen atoms, they are added into the oxaloacetate, and oxaloacetate is converted into malate. Then with the help of special transporter, which is malate alpha keto glutarate transporter, what happens that malate, it enters into the mitochondria through in a mitochondrial membrane. Now inside the mitochondria, what happens that malate, it is again oxidized into oxaloacetate. Now at this time, the enzyme which perform its function is mitochondrial malate dehydrogenase, and as a result of this activity, NADH is produced, and that NADH enters into the electron transport chain. Now, how this oxaloacetate, again it comes out of the cell. Oxaloacetate, it cannot cross the mitochondrial membrane, So what happens that oxaloacetate, with the help of mitochondrial aminotransferase, it combines with the glutarate to form aspartate and alpha ketoglutarate. Alpha ketoglutarate, it comes out through the malate alpha ketoglutarate transporter, and aspartate, it comes out from the mitochondria with the help of uh, aspartate glutamate transporter. Outside the mitochondria, again extra mitochondrial aminotransferase, with the help of extra mitochondrial aminotransferase alpha ketoglutarate and aspartate, they again combine to form oxaloacetate and glutamate. Glutamate will comes into the mitochondria, and oxaloacetate, it is again used to form malate. Now, second shuttle system is the glycerol 3 phosphate shuttle system. Glycerol 3 phosphate, which is produced extra mitochondrially into the cytoplasm as a result of the glycolysis. So, this glycerol 3 phosphate, it is it can cross the mitochondrial membrane directly, will enter into the mitochondrion, where it is oxidized to dihydroxy acetone phosphate with the help of enzyme glycerol 3 phosphate dehydrogenase. So it will deliver its electrons to FAD, and FAD is reduced to FADH. As a result of this activity, dihydroxy acetone phosphate is produced, which will directly crosses the mitochondrial membrane and will come out to again to be reduced by gaining the electron from NAD. So in this way, glycerol 3 phosphate. So glycerol 3 phosphate shuttle system is comparatively less complicated as compared to malate aspartate shuttle system. So now the question arises that how NADH will decide that it has to deliver electron through either malate aspartate shuttle system or glycerol 3 phosphate shuttle system. So it is tissue dependent process. There are some tissues which contain glycerol 3 phosphate shuttle systems, and there are some tissues which contain malate aspartate shuttle system. So it basically depends that which type of shuttle system is present in the tissue.